Hi, I'm Gulam Hussain Syed, scientist and principal investigator in the virus host interaction lab. My group works at deciphering this virus host interactions, how the viral proteins interact with the host proteins or the host organelles, and we are trying to identify therapeutic targets for developing antiviral agents. Viruses interact with the host cells or the host proteins from the time of the entry of the virus into the host cells till their egress. They interact with host organelles like mitochondria, cytoskeleton, etc. to exploit their functions and to create an intracellular environment conducive for the viral dissemination. My group characterizes these interactions between the viral proteins and the host proteins to develop host-targeted antiviral therapeutics with higher barrier against evolution of resistant mutants. Our focus is on arboviruses and flaviviruses like dengue, Japanese encephalitis, Zika, and hepatitis C virus that have emerged as formidable human pathogens.
My project is deal with the molecular diagnostic of risk 2 and risk 3 pathogens which are potential threat identified by ICMR. Additionally, I am dealing with the cellular death pathways that is induced by the flaviviruses.
When a virus primarily attacks our body, it tries to hijack our physiology for its own benefit. We are trying to understand how these viruses hijack our host machineries so that we can develop strategies and drugs to stop these interactions and eventually stop the spread of the virus. My work primarily focuses on virus-mediated neuroinflammation and neurodegeneration.
My research work primarily focuses on understanding how arboviruses utilize the host mitochondrial dynamics and exploit it for its own benefit. My key focus is on looking for inhibitory molecules that can perturb these interactions between virus and the mitochondria. My work focuses on characterizing the cytoskeletal modulation upon the virus infection. Here, this will further aid in identification of host-derived drug targets.
We utilize high-throughput and high-content imaging platforms to screen approved drug libraries for repurposing against viral infections. Additionally, we examine extracts from marine resources to identify novel antiviral agents and compounds that can modulate cellular stress. We aim to bioprospect new marine compounds to benefit human health. Our institute has all the high-end infrastructure and equipment to carry innovative and novel research